Welcome to the deep dive. Today we're jumping straight into the heart of genetics. We'll trace how our understanding exploded, really, from basic ideas to incredibly precise molecular tools. And we have this great chapter overview to guide us, but honestly, the current stuff is so revolutionary we have to start there. Let's talk about CRISPR-Cas. If you follow science at all, you've heard of it. It's basically the ultimate toolkit for editing genomes, human, plant, animal, you name it. The precision is just staggering compared to what came before.
Absolutely staggering.
Yeah.
And a big part of why it's taken off is its relative simplicity compared to older methods. It actually comes from bacteria. It's part of their immune system.
Mhm.
But for our purposes, the core idea is you design a small piece of RNA, the CRISPR RNA. You make it match the exact DNA sequence you want to target.
Right, so you're not building a whole complex protein from scratch for every target site. That was the issue with things like zinc fingers, EFNs, and talons, wasn't always custom engineering.
Exactly. That was the bottleneck.
Mm-hmm.
With CRISPR, the RNA is like a programmable GPS. It takes the Cas enzyme, Cas9 is the famous one, straight to the right spot in the genome. And then the Cas enzyme does the work, usually making a cut, which allows for editing. So this RNA-guided approach makes it, well, more accurate, definitely easier to use, and just incredibly versatile.
And the potential uses are just vast. We're looking at potentially fixing the root causes of genetic diseases, things like cystic fibrosis, Huntington's, muscular dystrophy.
The therapeutic potential is huge, but it goes beyond human health, too. Think about agriculture creating crops resistant to pests or drought.
Yeah.
Or even public health, like modifying mosquitoes to stop them spreading malaria.
Okay, let's pause there. This level of control, editing DNA like text, it feels futuristic, but it builds on centuries of discovery. We need to understand the foundations to really grasp CRISPR's significance. Let's trace that history.
Definitely. The very earliest genetics wasn't science as we know it, but practical application. Go back thousands of years, 8,000 to 1,000 BC, people were already doing selective breeding, domesticating plants and animals. They were shaping the genetic makeup of species without knowing the underlying rules, just by observing traits.
And the early philosophical ideas were interesting, like the Hippocratic idea of humor as carrying traits, or Aristotle thinking male, vital heat-shaped female substance. Lots of guessing.
Right, pre-scientific attempts to explain heredity. Things got more concrete much later, around the 17th and 19th centuries, with better biology. Key ideas like epigenesis, the organism developing step by step.
Instead of the old preformation idea that a tiny person, a homunculus, was already inside the egg or sperm.
Exactly. And critically, the cell theory, the realization that all life is made of cells, and cells come from other cells. That set the stage.
But the real game changer for understanding inheritance, what we call transmission genetics, was Gregor Mendel, 1866.
Yes, Mendel. His work was foundational. What's incredible is he worked out the basic rules of heredity quantitatively with his pea plants, long before anyone knew about DNA or chromosomes. He proposed that traits are controlled by pairs of factors, we call them genes now, and these factors segregate, they separate when organisms make gametes, like sperm and eggs.
It's kind of mind-blowing. He used math and careful observation. No microscope's powerful enough to see the physical stuff, just logic.
Pure genius. And it created this gap, right? His abstract factors worked mathematically, but what were they physically? That answer came with better microscopes. Scientists could finally see chromosomes inside cells. They noticed that most cells have two sets of chromosomes, the diploid number or two Ns.
Homologous pairs, one set from each parent.
Right. And crucially, when cells prepare gametes through meiosis, the chromosome number gets halved to the haploid number, N. Each gamete gets just one chromosome from each pair.
So, wait. The way chromosomes separated during meiosis, did that match how Mendel predicted his factors would separate?
It matched perfectly. That was the huge insight. Walter Sutton and Theodore Boveri independently put it together around 1902, the chromosome theory of inheritance. They basically said, look, these chromosomes behave exactly like Mendel's factors. The factors, the genes, must be located on the chromosomes.
That connection is fundamental. Genes are on chromosomes, and this led to studying variation, often in the fruit fly, Drosophila, easy to breed, lots of offspring.
Yes, Drosophila became a key model organism. Thomas at Morgan's lab made huge discoveries, like finding a fly with white eyes among the normal red-eyed population.
How spontaneous change.
A mutation, exactly.
Yeah.
A heritable change in the genetic material. That's the ultimate source of all variation.
And that white eye trait is caused by a specific version or allele of the eye color gene. Different alleles lead to different traits.
Precisely. So the specific alleles an organism has, like the white eye allele versus the red eye allele, that's its genotype.
And the observable trait, the actual eye color you see, that's the phenotype. Genotype determines phenotype.
Correct. So, okay, genes are on chromosomes. The next big question was, what part of the chromosome is the gene made of? What's the actual chemical carrying the information?
And for a while, the smart money was on proteins, wasn't it? Because proteins are complex, diverse molecules, DNA seemed, well, a bit boring chemically. Just 4 repeating units.
It did seem too simple to many. Proteins have 20 different amino acid building blocks, much more potential complexity, it seemed. But then came the critical experiments. Avery, McLeod, and McCarty in 1944 showed pretty definitively, using bacteria, that DNA was the transforming principle. It carried the genetic information.
But there was still some skepticism.
There was. The final nail in the coffin for the protein idea came from Hershey and Chase in 1952. They used viruses that infect bacteria, bacteriophages, and elegantly showed there was the viral DNA, not the protein code, that entered the bacteria and directed the production of new viruses.
So DNA is the stuff, which leads us right to 1953 and probably the most famous discovery in biology, the structure of DNA, Watson and Crick.
The double helix. Absolutely iconic. It wasn't just beautiful. The structure immediately suggested how DNA could do its job.
Describe it for us again. It's like a twisted ladder.
Exactly. 2 long strands, the backbone's made of sugar and phosphate, and the rungs of the ladder are pairs of nitrogenous bases. Adenine, guanine, thymine, cytosine A, G, T, C.
And the pairing is specific, right? A always pairs with T, and G always pairs with C, complementary base pairing.
That's the key. That complementarity explains how DNA can be copied so accurately. Each strand can serve as a template for making the other. It also showed how information could be encoded in the sequence of bases.
And RNA is similar, but slightly different.
Right. RNA usually uses the sugar ribose instead of deoxyribose. It uses uracil U instead of thymine T, and it's typically single-stranded, not a double helix.
Okay, so we have the structure. How does the information stored in DNA actually get used by the cell? This leads to the central dogma, right?
Yes, the central dogma of molecular biology. It describes the flow of genetic information. RNA makes protein.
Let's break that down. Step 1 is transcription.
Right. In eukaryotes, the DNA stays safe in the nucleus. A specific segment of DNA, a gene, is used as a template to synthesize a complementary strand of messenger RNA, or mRNA.
So you're making a working copy of the gene's instruction.
Exactly. Then that mRNA molecule travels out of the nucleus into the cytoplasm. Step 2 is translation.
Where the mRNA message is read to build a protein?
Correct. The mRNA docks with the ribosome, which is like the protein-building machinery. The ribosome reads the mRNA sequence in groups of 3 bases called codons.
And each codon specifies a particular amino acid.
Almost always, yes. There are 20 common amino acids. Another type of RNA, transfer RNA or tRNA, acts as the adapter. Each tRNA carries a specific amino acid and recognizes the corresponding codon on the mRNA.
So the ribosome moves along the mRNA, reading codons, and tRNA brings in the right amino acid, linking them together into a chain,
which folds up into a specific 3-dimensional shape, forming the final functional protein. And proteins do almost everything in the cell enzymes: structural components, signaling molecules.
like insulin or collagen.
Exactly. And the link between a tiny change in DNA and a big change in the organism is incredibly clear in diseases like sickle-cell anemia.
Ah, right. That's a powerful example. Remind us how that works.
It's caused by a mutation in the gene for hemoglobin, the protein that carries oxygen in red blood cells. Specifically, it affects the beta-globin chain.
And it's just one tiny change in the DNA.
One single nucleotide change. In the DNA template strand, a CTC sequence becomes K, just one T changing to an A.
Okay, so during transcription, that changes the mRNA codon.
Yes. The normal mRNA codon, GG, becomes GUG.
And J codes for one amino acid, GUG for another.
Right. GAG codes for glutamic acid. GUG codes for valine. So just one amino acid is swapped out of the 6th position in the entire beta-globin protein chain.
One amino acid difference out of hundreds.
Yes. But this single change has drastic consequences. The altered hemoglobin protein tends to clump together, to polymerize, especially when oxygen levels are low.
And that clumping distorts the red blood cells, makes them sickle-shaped.
Exactly. Those misshapen cells can't flow properly through small blood vessels. They block capillaries. This leads to pain, organ damage, anemia, all the symptoms of the disease. It's a stark illustration of genotype determining phenotype right down to the molecular level.
Wow. That really drives home the connection. Okay, so understanding DNA structure and the central dogma laid the groundwork for actually manipulating DNA.
Absolutely. The next huge leap came in the 1970s with the dawn of recombinant DNA technology, A key discovery was restriction enzymes.
You mentioned enzymes earlier. These are different.
Yes, these are enzymes found in bacteria that act like molecular scissors. They recognize specific short DNA sequences, maybe 4 to 6 base pairs long, and cut the DNA only at those specific sites.
Like a precision cutting tool. What were they for in bacteria?
Defense. They cut up the DNA of invading viruses. But researchers realized they could use these enzymes to cut DNA from any source in a predictable way. You could cut out a specific gene, for instance. Then you could paste that gene into a carrier molecule, often in a bacterial plasmid called a vector.
Creating a hybrid DNA molecule or recombinant DNA.
Precisely. Then you introduce this recombinant vector back into a host cell, usually bacteria like E. coli. As the bacteria multiply, they copy the vector along with their own DNA, creating millions of identical copies of your gene of interest. That's cloning.
And this ability to cut, paste, and clone DNA fragments basically launched the whole field of biotechnology.
It really did. Biotechnology is essentially using these molecular tools to create products. Think genetically modified crops, plants engineered to resist herbicides or pests. These are now widespread.
Or using genetically modified organisms to produce useful proteins.
Yes, like producing human insulin in bacteria for diabetics, or even generating therapeutic proteins in the milk of transgenic animals. Remember Dolly the sheep? That was an early famous example of cloning a whole mammal, pushing the boundaries.
And as these technologies got more powerful and automated, especially DNA sequencing, we moved beyond single genes to looking at everything at once.
Right. That led to the omics revolution.
Yeah.
Genomics, studying entire genomes, all the DNA in an organism. Proteomics, studying the full set of proteins produced. And bioinformatics, the crucial computational side, developing the databases and algorithms needed to store, analyze, and make sense of this overwhelming amount of data.
The Human Genome Project being the ultimate example of genomics and bioinformatics in action.
Absolutely. A massive undertaking that gave us the blueprint.
It feels like the way scientists ask questions has changed too.
Definitely. The traditional approach was classical or forward genetics. You'd find an organism with an interesting new trait, a phenotype, maybe caused by a random mutation. Then you'd do the hard work of figuring out which gene was responsible.
Working from the trait back to the gene.
Right. But now, especially with sequenced genomes, we can do reverse genetics. We can pick a gene whose sequence we know, but maybe we don't know its function. Then we can use techniques like gene knockout to specifically disable or knock out that gene in a model organism. Then we observe the organism to see what changes, what functions are lost. We figure out the phenotype after targeting the gene.
Working from the gene forwards to the trait. It's a completely different way of investigating.
It is. And both approaches rely heavily on model organisms.
Right, you mentioned fruit flies. Why use flies or worms or yeast or bacteria? Why not just study human genetics directly?
Well, practicality is a big part of it. Model organisms typically have short life cycles, they're easy and inexpensive to grow in the lab, and they produce lots of offspring, which is great for genetic studies. But the deeper reason is the fundamental unity of life. Because we all evolved from common ancestors, the basic molecular machinery of life is remarkably conserved.
Meaning a gene that does something important in yeast or a fly often has a counterpart, a very similar gene doing a similar job in humans.
Exactly. So discoveries made in these simpler systems often translate directly to understanding human biology and disease. For instance, studying how E. coli bacteria repair their DNA helped us understand genes involved in human colon cancer, like MLH1.
And Drosophila, the fruit fly, is used to study human nervous system disorders.
Yes, quite extensively. Many genes involved in human neurodegenerative diseases like Huntington's or certain forms of blindness like retinitis pigmentosa have direct counterparts in flies. Studying the fly version helps us figure out the disease mechanisms.
So these models are indispensable. But as we talk about gene editing, cloning, transgenic organisms, it brings us back to the ethics, doesn't it?
It absolutely does. The technology, especially things like CRISPR, is moving incredibly fast, often much faster than our ability to develop ethical guidelines, societal consensus, or legal frameworks to manage it.
What are some of the biggest concerns?
Well, a major one is the potential for modifying the human germline, making changes to sperm, eggs, or embryos that would be passed down to future generations. The implications are profound and, frankly, unpredictable.
Then there are issues around genetic discrimination. Could your genetic information be used against you for insurance or employment? And data privacy, who controls the massive amounts of genomic data being generated?
These are huge societal questions. There's actually been a significant international push to create some kind of global forum or regulations, particularly around those heritable human genome edits. The power of this technology demands really careful thought and public discussion.
So wrapping this up, it feels like we've covered the three main pillars of genetics. First, the basic rules of inheritance, transmission genetics, laid down by Mendel.
Right, how traits are passed on. Second, the nuts and bolts, molecular genetics, DNA structure, the central dogma, how genes actually work at a chemical level.
And third, the modern applications, biotechnology, genomics, using that knowledge to sequence, analyze, and even modify genomes.
Exactly. And, if you look at Nobel Prizes over the last several decades, genetics and molecular biology are consistently recognized. It just highlights how central this field is to understanding life itself. And the pace isn't slowing down.
So, you now have the framework, the historical context, and the molecular basis to really understand technologies like CRISPR and the discussions around them. Here's something to think about. We used to rely on chance mutations to learn about genes. Now, with tools like CRISPR, we can make almost any change we want, deliberately. How do we choose what changes to make? Which questions are the most important or the most ethical to pursue?
That's the core challenge, isn't it? Balancing the incredible potential to alleviate suffering and improve lives with the immense responsibility that comes with rewriting the code of life. It requires ongoing dialogue, not just among scientists, but within society as a whole.
Thank you for joining us on this deep dive into the foundations of genetics. We hope this gives you a solid base for understanding the rapid developments happening right now and encourages you to think critically about their impact.